a review of integumentary system models. So we'll look at the layers, the types of tissues, and then some of the structures that we see in here and compare them in two different models. First, the layers. The overall layer from here to here, this is all the epidermis. In our previous video, I expanded that and looked at the 4 to 5 layers of the epidermis, depending on where you're at. Epidermis. Then the rest of this layer, all the way down so we get to this adipose tissue, is all the dermis. And then where we see this adipose tissue below the dermis is the hypodermis. On this model, we took out the epidermis. So there is no epidermis on this model. It's showing you dermis until we get to the hypodermis. The reason why we took it out is showing these wavy ridges here. And so we'd have connections with the epidermis. It'll be epidermal ridges that connect with these dermal papillae. And these describing the hills and valleys is actually what contributes to our fingerprints or footprints. And it's like an interlocking mechanism of the fingers coming together of the dermis and the epidermis. When we look at the dermis, and I go inside, the first layer here, it's about 20% of all the dermis, and I teach it as just finger length running through the model. This is the papillary layer. So if I continue on this side, just this width here, the layer is papillary layer. Then the rest of all of this, discarding the structures, we'll learn the structures in the layer, but this entire section here, it's like the rest of my hand in width when you compare it on the model. This is all the reticular layer. So the dermis is 2 layers, papillary layer, and then the rest is reticular. Over here, papillary, and then reticular layer until I get to that adipose tissue. The adipose tissue is what makes up the hypodermis. Now we look at some of the structures inside. First, let's look at these structures here. They have these little heads and tails, a head and then a tail drop down. On this side, you can see a head and then a head and a tail. These are called tactile corpuscles or Meissner corpuscles, and they're for light touch. So I can feel like the wind or a spider running along me, or just like light little touch helps me to feel things like I'm grasping. Those are called tactile corpuscles. Versus the bigger structure here, this brown one here, and then these 2 yellow ones that kind of look like onions. These are called Pacinian corpuscles, also called lamellated corpuscles. These are for deeper touch. So remember we said one of our functions of the skin is to be able to have specialized sensations. And these sensations include these corpuscles, these collections that have a nervous function, so I can feel light touch and deep touch. When we look at the hairs inside, and I run up to the top, we can divide it into 2 sections. The root is when I have the hair inside the skin. As it leaves outside the skin, it's called the shaft. So 2 regions of the hair, shaft, and then going inside, root. What surrounds the hair are these little flowery glands called sebaceous glands. Sebaceous glands. So these pink structures here, and then they opened it up here. It goes around the hair. On this one, it's this purple structure here. This is secreting oily sensations that we call sebum. So I get that oily sensation on my hair, my skin. That's produced by sebaceous glands. And if you remember, there's 2 types of glands in the body, exocrine and endocrine. These are exocrine glands because they're secreting their product to a direct target, to the surface of the skin. Remember, these glands are specifically epithelial tissue. We call them glandular epithelium. In a previous video, we talked about their mode of secretion. How do exocrine glands secrete the product? This one goes through a process called holocrine secretion, where the cells burst and die, and the product is that oily sensation. So sebaceous glands. That was a lot to say just for the sebaceous glands. Eccrine or Merocrine sweat glands. I see here, this structure here, and you see the tube disappear into the tissue. Same here. This is an eccrine or Merocrine sweat gland. And then this eccrine sweat gland shows the tube going all the way up to the surface, because this is our normal perspiration or water perspiration, another type of exocrine gland. And this one goes through a process called exocytosis, another mode of secretion. On this side, this white structure with the tube coming up, and this white tube with the tube coming up is my eccrine sweat gland. Other structure to see is that muscle that runs with the hair. It's called an Arrector Pili muscle. Arrector, to make something erect. Pili referring to a hair. And it's a muscle. So it literally is a muscle. So I ask my students, what type of tissue is this? There's only 3 types of muscle. Cardiac, smooth, and skeletal. Cardiac, this is not my heart. Skeletal, this is not attached to a bone. So by process elimination, this is a smooth muscle. So it's an Arrector Pili muscle is the name of the structure. The type of tissue is smooth muscle. It erects the hair. It's supposed to trap heat. This is what causes goosebumps. So when I erect the skin and I tighten it, I close the hairs together. In theory, I trap the heat to help me maintain the heat in my body. So it's a temperature regulation, but we see that when we're contracting the skin to generate some energy and some heat. But when we watch something amazing or a movie or a sport, you get those goosebumps. When you get scared a lot, all of a sudden you may get goosebumps as well. It's a defense mechanism to provide that heat. So Arrector Pili muscle. So we talked about layers and structures and then some of our modes of secretion.